Plant tissue. A tissue is a group of cells performing some collective function is clear tissue. Tissues organize the body of plant for performing different function. Many types of tissues associate with each other to form tissue system. Vascular system as an example of tissue system. The tissues have following characteristics. 1. Tissues are composed of many cells. These cells may be of one type or different types. 2. The cells in a tissue are held by tight junctions. 3. All the cells in a tissue have similar organization. 4. All cells in the tissue are involved in same activities. Classification of tissues. There are two types of tissues. Simple tissues. The tissues with similar type of cells are called simpler tissues. Examples: parenchyma, collenchyma and sclerenchyma.
Compound tissues. The tissues with different types of cell are called compound tissues. Example: xylem and phloem. Xylem is composed of tracheal cell, vessel cell and parenchymatous cells. Phloem is composed of sieve cells, companion cells and parenchymatous cells. Parenchymatous simple tissues with cells having thin and elastic walls are called parenchyma. Parenchyma forms the main ground tissues. They are continuous throughout the body. They are present in root, stem and leaves.
Cell structure of parenchymatous cell. Parenchyma cells have active protoplast. They have prominent nucleus. These cells have thin primary and secondary elastic walls. Their pratishat sans are rachifly composed of cellulose. These cells are closely packed. But intercellular spaces are produced by the dissolution of middle lamella. These air spaces are recommend for exchange of gases. Parenchymas have prominent intercellular spaces. The protoplast of the adjacent cells is connected by plasmodesmata. Shapes of parenchyma cells. The parenchyma has different shapes. 1. Polyhedral. These cells are mostly polyhedral in shape. 2. Stellate. These parenchyma are cooned in the stems of plants. They have well-developed air spaces between them. 3. Elongated. Elongated parenchymas are found in the palisade tissue of leaf. 4. Lobed. Lobed parenchymas are found in spongy and palisade mesophyll tissues of some plants. Origin of parenchyma. Parenchyma are mostly primary in origin. But they are also produced as a result of secondary growth greater than the functions of parenchyma. Parenchyma cells are less specialized. But it performs major activities in the cell. 1. Meristematic. They resume meristematic activity under certain special conditions. These conditions are wound healing, regeneration, formation of adventitious roots and union of glands. 2. Photosynthesis. Some parenchymas are involved in photosynthesis. The parenchyma cells of mesophyll tissues of leaves are rich in chloroplast. They are called assimilatory parenchyma or collenchyma. 3. Storage. Parenchyma also store food in fruits and roots etc. They are called storage parenchyma. These parenchymas have many leucoplasts in their cells. These parenchyma stores starch, fats, oils and other granules. Storage parenchymas are common in the cortex of stem, root and seeds. 4. Ground tissues. Parenchyma acts as ground tissues in most of the tissues. Secretory and excretory structure. In certain cases, parenchyma acts as a secretor greater than an excretory structure. 6. Pigmented cells. Parench greater than ma cells of flowers and fruits contain chromoplast. It gives colors to petals and fruits. 7. Storage of water. The parenchyma cells of succulent plants store water. These cells are large with thin cell wall. They have thin layer of cytoplasm. These cells have large vacuoles. These vacuoles contain mucilaginous sap. This sap increases the water holding capacity of the cell. Collenchyma. Parenchyma cells with uneven angular thickening which supporting growing part of plant are called collenchyman. Collenchyma tissues form separate strands or continuous cylinder near the surface of the cortex, in petioles and along the vein of the leaves. Collenchyma tissues are absent in roots. Collenchyma tissues are also absent in stem and leaves of many monocot plants.
Cell structure and organization. Collenchyma cells have living protoplasts. They are simple tissues and contain single type of cells. The cells are elongated with pointed and prismatic ends. They are capable of further growth and divisions. Collenchyma tissues also contain chloroplasts in green organs. Collenchyma form complete cylinder in stem. Collenchyma tissues are present in the margin of leaves. Cell wall in collenchymatous cells.
Their thickened primary wall is non-lignified. It is mostly composed of cellulose and poor in pectic substance. Therefore, it is elastic in nature. Triangular thickenings are rich in cellulose. An additional layer of microfibrils is present inside the petiole. In some cases, the cell and petiole of collenchyma is sclerified. Sclerification occurs by the formation of lamella. The lamellae are rich in cellulose. Sometimes, these lamellae become lignified. Additional lamellae appear inside. It decreases the lumen of collenchymatous cells. Size and shape. Different collenchyma tissues have different sizes and shapes. Mostly thesis cells are elongated with tapering ends. The longest collenchyma tissues are present in the central position. The shorter collenchyma tissues are present in the periphery. Types of collenchyma. 1. Angular collenchyma. The thickenings in the wall of these cells are present in the angles of the cells. They are found in the petioles of some plants. 2. Lamellar collenchyma. The thickenings in these tissues are present on the tangential walls of the cell. These are present in the stem cortex of some plants. 3. Lacunar collenchyma. In these tissues, thickenings are present in with faces intercellular spaces. Lacunar collenchymas are prex, in the petiole of composite species. Sclerenchyma, the lignified tissues, which lack protoplast at maturity, are called sclerenchyma. They have thick secondary wall. These are main strengthening tissues of plant. Sclerenchyma cells are found in all parts of plant. They are present in both ground tissues and vascular tissues. Sclerenchyma has two types, fibers and sclerides. A. Fibers very long and narrow. Sclerenchymatous cells with tapering ends are fibers. The length of the fibers varies greatly. Fibers are found in all parts of the plants. They are present in stern, roots and leaves. Fibers are also present in xylem and phloem. They may form component of vascular tissues or they are present outside the vascular tissues. Types of fibers. There are three types of fibers: Xylem, bast and septate fibers. I. Xylem fibers or wood fibers. They are present in xylem. They form major part of the xylem. Sclerified fibers have three main types. Libiform fibers. These fibers have very thick wall and simple pits. They are longer than the tracheids of that plant. Tracheids. They have intermediate thickness between tracheids and libiform fibers. They have bordered pits. 8. Mucilaginous fibers. The innermost layer of these fibers is rich in cellulose. It has greater nature holding capacity. Therefore, these fibers absorb %8 and swell. These fibers are common in elastic U. 2. Bast fibers. These lids are present in phloem and cortex tissues. In cortex, they form uninterrupted hollow cylinders in thick ground tissues. In phloem, they form fiber sheath. 3. Septate fibers. These are found in both Xylem and phloem. Hairs have interremiceptor. These are found in septate hairs. Development of fibers. Fibers develop from different meristems like procambium, cambium and round meristem. Fibers also develop from parenchyma cells. The initials of the primary fiber appear earlier. They grow in length and form fibers. The secondary fibers develop in the fully grown tissues. Commercial fibers, phloem or bast fibers have great economic importance. These fibers give a commercial fiber. These fibers are used in rope and cloth making. So many important plants which give commercial fibers are hemp, jute, kenaf, flaxseed. Sonic commercial fibers are obtained from the leaf of monocot plants. These plants are Manila hemp, bowstring hemp, New Zealand hemp and pineapple fibers, beet sclerides.
The variable-shaped sclerenchymatous cells with strongly lignified wall having simple pits are called sclerids. Any non-fibrous sclerenchymatous cell is elicit. Sclerids are found in different parts of plants. -1 They are present in epidermis, ground tissues and vascular tissues. They are present in the form of hard mass of cells. The seed coats of many seeds are entirely composed of sclerids. Sonic non-functional parenchyma cells are present in cuscular tissues. The walls of these parenchyma cells become thick and they become sclerids. There are 4 types of sclerids. 1. Ibracosclerids or stone cells. They have isodiametric shape. They are found in phloem, cortex, bark of stem and fruits. 2. Macrosclerids. They are rod-shaped. They are found in tester of seeds. 3. Astrosclerids. They are star-shaped. They are mainly found in leaves. 4. Osteosclerids. They are horn or spoon-shaped. They are present in seed coat. Development of sclerids. The development of sclerid is coordinate and intrusive. Coordinated. In the Cs, The cell wall continues to grow uniformly. This uniform growth on all sides without separation from neighboring cells is called coordinated growth.